Welcome to my world. Another day, another time, another period to acquire knowledge. Knowledge is power, health is wealth. Today we shall try to demystify the problems with drugs and addictions. The laboratory is the only place where you demystify most of this mystery. And one aspect of the medical laboratory that really demystifies such mysteries is the histopathology or histology and cytology department. And I shall now do some exposition on the brain to see the effects of the codeine on the brain. And this is not mere talking, because physically you can say, oh, it's affecting the man's brain, every problem is affecting the brain. But when we take a cross-sectional dissection of that brain itself, we can now see what has really deviated, what morphology has been affected, and some parameters, biochemical parameters, that has really shifted, or whatever has happened, we'll be able to expose. This is my effort today. If you like what you've seen, press the like button. Subscribe, and you'll be happy when you are notified of the next production. Laboratory finding in codeine addiction. This is the topic of interest today. We just want to unravel what really caused this abuse, as well as the effect on our children, especially those who are hooked on it, addicted to codeine. The outline. In introduction, cross-section of the brain, sample preparation, laboratory test and finding, and finally conclusion. The introduction. Drug abuse may be defined as the arbitrary over-dependence or misuse of drug with or without a prior medical diagnosis from qualified health practitioners. Referred. The misuse, abuse, and dependence of codeine products purchased over the counter is now public health challenge throughout the super. That's 20 from this. Codeine is also known as 3-methyl morphine. It is a phenylephrine derivative extracted from opium or synthetically produced by the methylation of morphine. That's why it is 3-methyl at that level, at the third level. It's one of the most commonly taken opioid medication at the center of opioid addiction problem in most countries, including the United States. Its addictive potential put it at risk of abuse. In addition to pain relief, codeine in higher doses can lead to a pleasurable euphoric sensation. When opiates enter the central nervous system, that is the brain, they activate the reward system of the brain, allowing for excessive release of pleasurable hormones, dopamine. It is this feeling that codeine addicts craved for. Addiction is a chronic dysfunction of the brain system that involves reward, motivation, and memory. Now, the effect on the human brain. Let's look at the brain itself. This is the human brain. You find the meninges, that's the covering, the skull, and all the other aspects. From here, it's the brain. These are all important parts. The brain stem is here. This is the cerebrum, divided into the right, left, and even the central hemisphere. And this, the midbrain pond, medulla and oblongata, constitute the level of the midbrain, and the cerebellum, which is called the small brain. So when you want to divide it into 3 compartments, that is what you have. You have the midbrain, pond and oblongata, medulla oblongata, and then the spinal cord. So these are the cells that makes up the cross section that we are talking about. Now, when you now look at the level of the cells, these are the dendrites, the neurons, and the neuroglia. So all these microglia are used for removing infection content, and this is a cross section of the brain. This is another cross-sectional view of the brain that you now see the Purkinje cells, the granular, so the parameter. This is a short cross-section, and now this is where you look at especially the cerebellum. You can see these are the brain neurons that really pass on the messages that sent to other parts of the body. Simple sample preparation. At the end of macro experiment, that is when we have taken the animals, separated them to you have the control, and what you do to control is probably you give the saline. The others you now grade the codeine concentration so that you can see the effects on the brain. The animals were now anesthetized with ketamine and intravenously injected after 12 hours overnight fast. The brain was harvested and weighed. After weighing, the brain was carefully divided into two equal hemispheres. One hemisphere was homogenized in phosphate buffer with a glass homogenizer, and the other hemisphere was used for histopathological analysis. That's the one that will tell us the effects on the internal structure of the organism. The homogenate was used to determine acetylcholine esterase activity, oxidative stress markers to check for the free radicals, and also the markers of genotoxicity, that is the one that will really affect the genes, the DNA itself. Histological sample preparation, that is those ones that we want to see the architecture of the cells, whether they are still intact. The brain from each rabbit was fixed in 10% formal cellular and processed for paraffin wax embedding. The paraffin embedded samples were sectioned at 3 micro and stained with H&E. Slides were examined using a light microscope for histological examination. These days, the electron microscope are available that could even do better work and even stain this thing so that we can see the internal organelles very clearly. Biochemical assay. Acetylcholine esterase activity was determined by colorimetric method. Markers of oxidative stress, neutrophil inflammation, and enzyme antioxidant were assayed using standard enzymatic colorimetry. The concentration of hydrogen peroxide, the marker for this thing, was determined by the method of use. The marker AGE, a marker of oxidative protein denaturing, and other oxidative, the 8-OHG, marker of oxidative DNA damage was determined using ELISA test kit. Lab tests and findings. The cerebral cortex of the high-dose codeine-treated animals showed normal flooring, molecular cell layer, and granular layer, but the area is the Purkinje cells layer showed mild depletion of blood. Codeine exposure in male rabbit led to a significant decrease in acetylcholine esterase activity. Low-dose codeine treatment significantly reduced acetylcholine activity when compared to the vehicle-treated control group by 25. High-dose codeine treatment caused a marked decline when compared. So you can know. This is a detailed labeling of the Purkinje cells, and these are contained in the hippocampus chamber, the component part of the brain. Hippocampus is a complex brain structure embedded deep into temporal lobes. It has a major role in learning and memory. So you can see when it is affected by whatever substance, it really affects the memory. And this is a picture that really shows the Purkinje cells. You can see the control still contains the Purkinje cells, but in the ones that we are testing for, the Purkinje cells have been depleted. You cannot see them, and that tells you so much about the effect on the Purkinje cells. The laboratory diagnosis. The assay of GPX is based on the oxidation of GSH to produce GSSG as part of the reaction in which it reduces cumin hydroperoxide. Glutathione reductase, GR, then reduces the GSSG to produce GSH, and the same reaction consumes NADPH. The decrease of NADPH measured at optical density a quarter of 340 nanometer is proportional to GPX activity. There is SH2, a marker of oxidative protein denaturation, then the 8-OHDG, the hydrogenase. The conclusion: high dose of codeine cause depletion of neuronal and punctual cells with reduced acetylcholine esterase activity. The acetylcholine activity involves activating the acetylcholine, which is very important in synaptic connection so that there is no dysfunction. So the messages will continue perfectly along to the appropriate peripheral nerves. The reactive oxygen species, we are not talking of the free radicals. All the free radicals that includes the hydroxy radical and the hydrogen peroxide were generated at high level, inducing cellular damage and cell death. These cell deaths was also proven to involve the induce apoptosis. We do apoptosis as cell death as well, the process of cell death through the 8-OHDG, which is an established biomarker of oxidative DNA damage, GNO. So when that happens, you know it has affected the DNA. Since oxidative stress can stimulate inflammation and vice versa, the observed raised MPO activity may be a result of codeine-induced oxidative stress or the other way around. The high-level free radicals, that's the conclusion in the main, created the depletion of neuronal and Purkinje cells. All the cells of high longevity of over 100 years and are irreparable are warning signs, warning of their consequences of future neurological diseases of Parkinson's disease, Alzheimer's, amyotrophic lateral sclerosis, multiple sclerosis, depression, and this is the reference, some reference.